in this BRIC seminar series. And I won't make again the biopic of Olivier that you were able to read in the announcement, but as you may know or not, Olivier is a medical doctor to begin. He's a pediatric oncology. But then very early on during his career, he was attracted by research, and then he entered INSERM. He was recruited at INSERM. Then he was in the group of Gilles Thomas, the Curie Institute. But then quickly he set up his own research unit, and he was very successful in discovering a number of oncogenes, a tumor suppressor, the Ewing sarcoma gene that he will discuss about today, but also he contributed to the discovery of the neurofibromatosis type 2 tumor suppressor, the rhabdoid tumor suppressor, together with our friend Nicolas, and then also the mutations of ALK in neuroblastoma, etc. And so he's a very fantastic researcher, very inspiring scientist and doctor. So it's a really great pleasure and honor. Thank you very much, Olivier, for coming.
Okay, thank you very much, David, for this very kind invitation and for the many different discussions I had all over the day today. It was great to meet PIs and to meet the students with very interesting questions and very interesting projects. And today I will speak mostly about Ewing sarcoma, which is a tumor which has kept us busy for the past few years. Let's say 30 or 35 years. And we are still quite far from really understanding what's going on, and far, maybe not so far, but not yet to have appropriate new treatments for the disease. So I will try to manage this complex. Yes. So just a few introduction on Ewing sarcoma, which was described by this guy, James Ewing, about a century ago in 1921, 102 years ago. It is mostly a tumor in the bone, observed in adolescent and younger adults. You see the mean age of diagnosis is around 15. And there is still a huge unmet clinical need for this disease, particularly for cases with metastasis. And as you can see, a metastatic Ewing patient nowadays cannot be efficiently treated. Localized disease have quite a good survival, around 75 to 80%. And two French cytogeneticists, Alain Orias and Claude Turcarel, in 40 years ago, so in 1984, described a very specific translocation in this disease between chromosome 11 and chromosome 22. And at the time of my PhD thesis, for a few years outside the medical field, the challenge was to know what was going on at the translocation breakpoint. So it took quite a few years with my colleagues to clone this translocation and to show that at the level of the translocation breakpoint, two genes were fused. We come back to those genes, one EWSR1 and the other one FLY1. And this is interesting because when I present this work, it took approximately 4 or 5 years for 4 persons to do this job 40 years ago. And now you can do it overnight. You just do RNA-seq and come back in the morning. Oh, yes, it's similar to that fly. Fantastic. So this is for the young student, that what you are doing now, things that maybe in a few years, it will be very easy. But thanks to you, the work that you have done for this. So this is one translocation among the many different translocations that can be observed, mostly in soft tissue sarcoma, mostly in young children, but not only. And so EWS-FLY somehow is a paradigm for all those translocations that can be found in solid tumors, and more particularly sarcoma. And when looking at this number of translocations and how many of them have really led to therapeutic progress, it's quite a few of them. And mostly only those that involve tyrosine kinase have led to fantastic clinical results, as for instance for the NTRK fusions in some sarcoma in kids and also in adult or ALK fusion. But for the many other fusions, yet there are not significant therapeutic progress made from the understanding of the genetics of the disease. So to come back to EWS Fly1, so EWS, which is now the name has changed, it was called EWS by us, but then the nomenclature called it EWSR1. So sometimes I will say EWS and sometimes EWSR1, but this is the same guy. It was named EWS because of Ewing sarcoma. It was the first time this gene was cloned, and it was named because it was cloned from the Ewing sarcoma. So EWS is an RNA binding protein by this, it's C-terminal half with this RRM, RNA recognition motif and zinc finger. It is a ubiquitous protein, and the real function of EWS is not very clear now. Fly1 is a transcription factor of the ETS family. So you may know that the ETS family of transcription factor has approximately 30 different members that play a very important role in differentiation and development. And so Fly1 is one of them. And it was named Fly1 because at the time we cloned the translocation breakpoint, it was already known in the mouse, and it was known as the site of integration of the friend leukemia virus. This name, friend leukemia integration 1, which is Fly1. And so as a result of the translocation, you have this EWS, EWSR1 Fly1. I will call it EF1 for simplicity reason. You have the DNA binding domain, which is linked to the N-terminal domain of EWS. And this N-terminal domain belongs to a large family of domains, which are called intrinsically disordered domain, prion-like domain, low complexity domain. And the specificity of those domains is that they can undergo liquid phase transition, just meaning that when the concentration of those domains increase in a cell or in a tube, at some point you will have formation of this kind of droplets, like oil droplets in water. And you will see that this property of EWS Fly, of EWS, is important to understand the function of EWS Fly. So EWS Fly, EF1, combine two types of motif. One is the classical ETSM. It is very complex to play with this. Wow. That doesn't work with that. Well, anyway, well, I'm on it. I don't move. So this is a single GGAA with a few flanking nucleotides, which is the consensus motif for the whole ETS family. And what we think EWS Fly is doing, and there are many experiments on this, is that EWS Fly is mostly competing with wild-type ETS factor on this single GGAA motif. The other motif is much more specific for EWS Fly. This is a repeat of this GGA motif, which is called a microsatellite sequence, and none of the wild-type X protein can bind to this kind of motif. Only EWS fly can bind to this. And the more GGAA you have, the more EWS fly can bind to this motif until a maximum of approximately 20 GGAA repeats. And so this is a complete neomorphic function of EWS fly, not observed in any wild-type protein. And this is a gain of function that for EWS fly. The idea that we have on the model to explain what EWS fly is doing, that this kind of motif, usually in normal cell, is completely transcriptionally silent. It is not known any normal role for this microsatellite in the genome, and no protein can bind to, have been shown to bind to this. Upon EWS fly expression, you have EWS fly able to bind to this microsatellite, which leads to an increased concentration, local increase of the concentration of the EWS domain, low complexity domain, I told you about it. And this low complexity domain it leads to a phase transition, which can lead to homotypic and heterotypic interaction with other proteins having this kind of low complexity domain, and particularly the transcription machinery, the chromatin remodeling machinery, and so on, leading to, for this silent, normally silent microsatellite to become active neo-enhancer, meaning that EWS fly leads all over the genome to the formation of 100,000 of new enhancers. Just an example of this. So you have here EWS fly binding, a peak. This is a ChIP-seq, a peak of EWS fly, active histone mark for enhancer, H3K4 methyl 1 and H3K27 acetylated. So in each, almost each of the microsatellites in the Ewing genome, you have the formation of this kind of enhancer. And this leads to a complete reprogramming of the cell of origin. The cell of origin has been a question for many years for Ewing sarcoma, because some people suggested it was epithelial, it was mesenchymal, it was endothelial. It was the initial idea of James Ewing. I will come to this a bit later in my talk, but this is understandable because EWS fly completely reprogram the genome of the cells of origin. This may be also one of the reasons why EWS fly is so toxic. We were speaking with some of you with NFAT-C2, EWS NFAT-C2. We don't know exactly how EWS NFAT-C2 is working, but EWS fly, you know, in the genome, it completely transforms the genome, and probably very few cells can really tolerate this huge change of EWS fly. With this in mind, I would like to address some of the questions about Ewing sarcoma that we have addressed across the past years. And also showing the role of these microsatellites in the development of Ewing sarcoma. First aspect I would like to develop is the genetic susceptibility. And this has been described 50 years ago by Joe Fromeni, who is the same guy that described the Lee and Fromeni syndrome, showing that the epidemiological profile of Ewing sarcoma was quite strange, because Ewing sarcoma was mostly observed in population of Caucasian origin, European origin, and quite rare in population of African origin, and kind of intermediate in population of Asian origin. And this has been quite investigated in depth recently by Logan Spector and Josh Schiffman showing the distribution of Ewing sarcoma worldwide. And you see that most of the Ewing sarcoma are observed in population of European origin. Even in Australia, it's known that Australia mostly of European origin. Even though in Africa, the epidemiological knowledge is not as developed as in European countries, Ewing sarcoma is very rare in Africa. And this is particularly visible in the US, so in the same environment. You have on the top the white population, so European descent, and on the bottom you have the black population, and you have approximately a 10 times difference between African origin and European origin. And this led us to interrogate this susceptibility. I'm saying susceptibility because you all know that a lot of cancer predisposition syndrome. And for Ewing sarcoma, it's not a cancer predisposition. There are very, very few families with Ewing sarcoma. So classical ways of investigating predisposition through pedigrees cannot be adapted to Ewing sarcoma. This is why we used genome-wide association study. So what you do when you do a genome-wide association study is that you try to have the maximum number of germline DNA from Ewing sarcoma patients and controls, and you analyze those sets of DNA with hundreds of thousands of different SNPs. And what you are looking is there a region in the genome where the frequency of the SNP differs between Ewing sarcoma patients and control patients? Here, the control patients obviously are of European origin, because if you do the same thing between Ewing sarcoma patient and African, you will have many confounding factors apart from Ewing sarcoma, obviously. So you try to have two populations match as much as possible with only one difference, and the difference is Ewing sarcoma or not Ewing. And you see a number of peaks with different SNPs in a region whose frequency is highly different between Ewing sarcoma patients and control. And these SNPs correspond to haplotype, so a region of the genome that was inherited as a block, and with all the SNP on this region showing up in this differential analysis. And what I just want to show you with this is that at each of those 6 loci, we have polymorphism in microsatellite sequences. And I will show you one example with the chromosome 10. So in the middle, you see a very strong peak. And just to explain you this, the y-axis is the inverse of the p-value. So here you see the 10, the p-value is 10 to the minus 16. So it's very significant. There's clearly something different here between Ewing sarcoma patients and control. And at this level, on chromosome T, you have an important gene, which is EGR2, also known as CROX20, which is a transcription factor. And Thomas Grunwald, who was a postdoc in the lab at this time, to show that EGR2 was an important gene in Ewing sarcoma, used a Ewing cell line and introduced an EGR2 small hairpin RNA inducible by doxycycline to inhibit EGR2, showed on the Western blots on the top. And you see that when he left the tumor grow for a while, and then added doxycycline in the drinking water of the mice. And as you can see with the red curve, when inhibiting EGR2, the tumor completely regressed. So showing that EGR2 is a very important gene in Ewing sarcoma, and that without EGR2, Ewing sarcoma cannot grow. And he did quite a lot of work to try to figure out what was going on in this region. And what he found a microsatellite a GGA repeat here, you see. In the non-risk allele, you see that this microsatellite is interrupted by a GGAT. The GGAT would not bind EWS5. And in the risk allele, what you have is just a single nucleotide change. You have a GGAT change to the GGAA. And so you change the 11 GGA stretch into 16 GGA stretch. And you can see on the bottom that the expression level of EGR2 is dependent upon this genotype, since the AA allele, so the GGAA allele, have expression level of EGR2 very significantly higher than GGAT, that the activation on a reporter system with the 16 as compared to the 11 leads to much higher increase of expression, and that EWS fly can bind much more efficiently to the GGAA allele than to the GGAT allele. So showing that possibly just a single substitution, base substitution in a microsatellite impacts on the efficiency of EWS5 to activate EGR2 and then to transform the cell. And we call this transformation facilitating polymorphism. And we have also a very similar example. So what is interesting also, because why we did the GWAS is like to find important gene for Ewing sarcoma and always potentially target for immunotherapy. So EGR2 is a transcription factor, so it's not an easy target. But what is interesting is that EGR2 regulates the mevalonate pathway, which is essential to synthesize cholesterol in the cell. And they are very good inhibitors of the mevalonate pathway. I'm sure that many of us are treated with this. These are statin. And it's very interesting that Ewing sarcoma linked to this has a very exquisite sensitivity to statin. So another example of the role of this polymorphism of GGA repeat is another locus on chromosome 6. And again, what we observe on the bottom here, that there is a strong difference in the length of the microsatellite between Ewing patient and control patient, much higher expression of RREV1 in those samples. And we did also some IC and iChip experiments showing that there is indeed a connection between the microsatellite, an answer linked to EWS-fly at the microsatellite, and the promoter of RREB1. And RREB1 also, I'm always trying to do that with this, but RREB1 is also very important for Ewing sarcoma, and knocking down RREB1 impact on the growth of Ewing sarcoma and clonogenicity of the cells. So we are still investigating the other loci, and we have some work being done on the chromosome 1, which is also very strong susceptibility locus for Ewing sarcoma. And again, we find polymorphism in microsatellite sequences. So this highlights, I think, the importance of this. And another aspect that is also potentially interesting for therapeutic aspect is that both EGR2 and RREB1 are two transcription factors that are downstream of the RAS-MAP kinase pathway. And indeed, it has been shown that in some cases, not all, unfortunately, but Ewing sarcoma may respond to inhibition of the RAS-MAP kinase pathway. Unfortunately, the cell responds very transiently and then can adapt to the escape to this inhibition. So I mentioned the cell of origin, and the way we became interested in this was through observation of the phenotype of Ewing cells when EWS-fly is knocked down by siRNA or small elfin RNA. And it was very striking that the cytoskeleton is completely disorganized in Ewing sarcoma, shown on the left. But when EWS-Y is inhibited, you have a much more fibroblastic organization of the cytoskeleton, as shown on the right. And so we dug a bit on this, and we showed, it was the work of Frank Tirod when he was a postdoc in the lab, we showed that if you take Ewing sarcoma, you inhibit EWS-Y, and you add also some differentiation cocktails to the adipogenic or osteogenic lineages, then when you inhibit EWS-Y, those cells can differentiate into adipogenic or osteogenic lineage. Therefore, strongly suggesting again that Ewing sarcoma, where the cell of origin was some kind of mesenchymal progenitor of mesenchymal stem cells. And so this is, I told a few about this later today, why we decided to try to transform normal human mesenchymal stem cells with EWS-Y to see, can we recapitulate the phenotype? And for this, taking into account the potential polymorphism that may help EWS-Y to have its action, we started from mesenchymal stem cells from a Ewing patient. So mesenchymal stem cells were sampled at the same time of the surgery for a Ewing sarcoma patient. And we used those cells, and by CRISPR-Cas9, introduced the translocation. And also, I will come to this a bit later, added a STAC2 mutation, which can be observed in Ewing sarcoma. And indeed, when we do this, we have an epigenetic profile, which is completely similar to the one of Ewing sarcoma. You have here, I will try with this. This is really a challenge. So here you have the ChIP-seq Fly1 signal. On the left, on A673 with the Ewing cell line. In the middle, in mesenchymal stem cells, you don't have the EWS-Y signal, obviously. And those mesenchymal stem cells transform with EWS-Y, you recapitulate exactly the same profile. And when you look at different loci, you have very similar profile. And even more importantly, if you look at the phenotype, I know there are pathologists in the room, but the phenotype of the tumor in mice, or injecting the cells in mice, is very similar to the one of Ewing sarcoma, and particularly with a strong membrane CD99 staining. So, and also something important with respect to the mesenchymal stem cell origin is that when you take Ewing cells and use an inducible small hairpin RNA to decrease EWS-Y, and you make this kind of nodules with the cells, and you observe those nodules. So I'm not sure the video will start. I don't know. No. No. The video will not work. Oula. I'm lost.
Yes.
Oui, mais là, c'est pas grave si la vidéo passe pas. I just wanted to show you that. Peut-être il faut y aller là.
Ouais, et c'est où la vidéo?
Normalement, tu as un truc qui est là, mais là je ne le vois pas. Mais peut-être qu'il faut que tu enlèves le laser.
Bah, c'est l'équivalent de la flèche.
Ouais, bon, il n'y a pas de vidéo. Mais juste que vous montrez, c'est dommage, c'était assez joli. C'est quand il y a EWS-Y, vous voyez les nodules grossir de façon homogène, sans cellules qui sortent du nodule. Par contre, quand vous regardez en inhibant EWS-Y, le nodule ne grossit pas, parce que sans EWS-Y, les cellules ne poussent quasiment plus. Mais par contre, les cellules se mettent à migrer en dehors du nodule comme extrêmement fort. Et donc ça, I'm sorry. I think I switched. Thank you. Okay, sorry for this. So this led us to the idea that is EWS fly activity homogeneous in a tumor? Or is there some heterogeneity of EWS fly activity with EWS fly I cells having a strong proliferation potential and EWS fly low cells having a strong migration potential? And would this balance between EWS fly and EWS fly I and EWS fly low cells be important for Ewing sarcoma? And so what we did is to do a single cell analysis of Ewing sarcoma. And what is shown here is to investigate those single cells with EWS fly activity signature, which is in red, strong EWS fly activity signature, and blue, low EWS fly activity signature. And you see that you have a very strong heterogeneity in those Ewing cells, with some cells having a very strong EWS fly activity, where also other cells have a low EWS fly activity. So we can map also G2M and G1S signature. And you see that G2M and G1S are not the cells with the highest signature, but mostly the cells with intermediate signature. So this is interesting. You have cells with very high EWS fly do not really proliferate, do not have active cell cycle. Cell with a high, but not very high EWS fly activity proliferate, and cells without EWS fly, with a low activity of EWS fly, mostly do not proliferate, but have a strong migration signature. Well, this is also other signature. You see that EWS I cells have a strong OXPHOS signature. As in the contrary, cells with lower EWS fly signature have mostly a glucose catabolism. So this led us to this model that we have, that in Ewing cells, you have heterogeneity of EWS fly activity. Some cells with a low activity of EWS fly recapitulate their mesenchymal features somehow of their cell of origin, can migrate, have a low proliferation rate, and those cells potentially can migrate in different tissue, in different environment. They have a very strong cell matrix adhesion properties, and so they can attach to the matrix, and then potentially reacquire a strong activity of EWS fly and lead to the formation of a metastatic site. And this is something that we investigated in samples. And indeed, in tumor samples, we could identify cells with a low EWS fly activity, human Ewing samples, and show in the mouse that if we sort cells with a low activity as compared to cells with a high EWS fly activity, we have much more metastasis with a low activity than with a high activity. So we believe that this plasticity of the cells between EWS fly high activity and low activity is something probably very important in the biology of the tumor and in the resistance to treatment, because EWS fly low cells are mostly resistant to treatment. So chemotherapy will eliminate cells, cycling cells, so EWS fly high mostly cells, but potentially keep the EWS fly low cells, which can then regrow as EWS fly. Okay, so we believe that this plasticity is important and needs to take into account two aspects. One is the cell of origin, which is mesenchymal stem cells, with all the characteristics of the mesenchymal stem cell, particularly the migration properties. And the other one is the key EWS fly target to grow as metastatic site. So our current model for Ewing sarcoma is that the cell of origin is some kind of mesenchymal stem cells. Mesenchymal stem cells is a bit still a fuzzy concept. It's not very clear what kind of cell it is. But this is one type of mesenchymal stem cells. This type of mesenchymal cells is transformed by EWS fly, which activity is facilitated by transformation facilitating polymorphism, particularly in microsatellites, and that then the cells can shift between high and low EWS fly activity, which is probably very important for the plasticity resistance to treatment. And the reason why EWS fly is shifting its activity are probably multiple. What is clearly known is that hypoxia involves this. If you have acute hypoxia, you put Ewing cell into acute hypoxia, you will have increase of EWS fly. But if you leave the cell in hypoxia for a few days, then you will have a strong decrease in EWS fly expression and activity. So EWS fly is the main driver of EWS fly, but in some cases it does not act alone. One of the most frequently mutated gene in Ewing sarcoma is STAC2. STAC2 is a member of the cohesin complex and can leads to loops in the DNA. And Didier Surdet, who was a postdoc in the lab, did a wonderful job on this, on STAC2 mutation, generating isogenic STAC2 Ewing cells and doing a lot of I-chip and many different looking at chromatin aspect. And what he observed is that at this neo-enhancer I showed you in microsatellite, when STAC2 is mutated, you have some kind of relaxation of the interaction between neo-enhancer and promoters. And this leads somehow to an EWS fly low phenotype. And the mutation of STAC2 is associated in Ewing sarcoma with metastasis and with resistance to treatment. And the idea that is behind is that when you have this mutation, inactivating mutation of STAC2, you lose STAC2, so you relax somehow promoter enhancer interaction, so you decrease EWS fly activity, and you increase potentially the migration potential. So our model is still the same, but with STAC2 loss of function that pushes a bit on the mesenchymal phenotype, increased metastasis and resistance to chemo. Yeah, I still have. I would like to shift completely now and come to what was shown in my title, immunology. What we observed, and this was very fantastic, two fantastic PhD students, Julien Vibert and Olivier Delattre. They could show that some of these microsatellites are obviously outside of any known gene, in gene desert. And in gene desert, those microsatellites have attract with CWS fly histone activating marks and lead to transcription of intergenic, otherwise completely intergenic region. As shown here, this is a gene desert, and you have EWS fly, you have these genes that show up with different exons. And when EWS fly is inhibited, you completely lose this gene. And this is another example here of this kind of gene. And what is quite interesting is that those genes are induced by EWS fly, so they are not expressed in any normal tissue which do not have EWS fly, and they are not expressed in any tumor tissue. tissue either. This is the plot on the whole TCGA dataset and on the whole GTEx dataset, and you see in red, these are the Ewing sarcoma, and you see very little expression outside of this. And this is another way. This is another way of looking at this with a heat map, and you have in red the 26 here neogenes, what we call neogenes, or genes, unknown genes that are expressed in Ewing sarcoma and not in any normal tissues and in any tumor tissues. Those neogenes have features of long intergenic non-coding RNA that are multi-exonic, more than 200 base pair long. And this is another example of the regulation of those neogenes. So this is single molecule RNA fish. On the left, in green, you have neogenes, and in red, the control genes. And you see when you inhibit EWS5, you completely lose the green signal and do not affect the red signal. Those genes are regulated by microsatellites. So if we do some CRISPR-I experiment on those microsatellites, we completely lose the expression of those genes, which this model, so in non-Ewing cell or in the parental cells, you have the microsatellites that are completely silent. Their role is not known. In Ewing sarcoma, you have EWS5 able to bind to this microsatellite to act as, to transform them into an answer, able to activate here, so for example, gene B through looping of chromatin, but also able to activate intergenic region. And so this long intergenic non-coding RNA, there are more and more publications showing that what is called link non-coding RNA may not be as non-coding as their name would suggest. And there are a number of publications showing that proteins, peptides, encoded by those long non-coding RNA can be observed in Ewing cells. Not in Ewing cells, in cells. And so we therefore speculated that if those neogenes induced by EWS1 encode proteins or peptides, this could be fantastic immunotherapy targets because they are completely specific for the tumor cells. And they are tumor specific, they are shared across all Ewing sarcoma because they all have EWS5, and they are directly linked to the oncogenic process because this is EWS5. So we did a lot of analysis to look if those neogenes indeed encode transcript, ribosome profiling to see whether we can find those neogene attached to a ribosome with looking at ribosome protected fragment. We did some whole cell proteomic and also immunopeptidomic. And indeed, for some of those genes, we could have a very strong indication by the ribosec that indeed there is some ribosec signal as shown here, and by whole cell proteomic that indeed we can find the protein corresponding to the expected open reading frame in those genes. And this was done by Céline, and a PhD student, and Florian, also a fantastic postdoc. So with this, unfortunately, none of those neoproteins encoded by those neogenes were at the surface of the protein, because they would have been fantastic target for CAR T cells if we could have something like this. But we could not yet find any of those proteins at the surface. And so we think, can we find peptides corresponding to those neoproteins attached to HLA class 1 molecules? And so we did a lot of immunopeptidomics on Ewing cell lines and PDX, and could indeed find peptide at the right side, so around 9 mers for peptide attached to class 1, corresponding to the appropriate HLA type of the cell lines. And for immunopeptidomic, for the different NGs, you see that we could find a number of peptides corresponding to these neogenes, with, as you can see, one of those neogenes, for any reasons, neogene 8, NG8, having a number of different peptides that can be bound to HLA. And so we said, well, if we have peptides corresponding to this peptide, if we have peptide corresponding to these neogenes, so could they be target for immunotherapy? Would specific T cells be able to recognize this peptide in the HLA context and be able potentially to attack Ewing cells? So we started from LC controls, isolated the CD8 cytotoxic T cells, isolated by tetramer staining the CD8 that are able to recognize the peptide from the neogene, as shown on the left. So we could isolate T cells, recognizing this peptide, expand those T cells in vitro, and then begin to test with Ewing cells. First test, what if we incubate those T cells with Ewing cells, are Ewing cells able to activate those T cells to increase cytokine production? And indeed, this is the case, but this is the case only with Ewing cell line that have the appropriate HLA type. We started with HLA-A2, which is the most frequent type in the human population. And you see in blue, these are non-HLA-A2 Ewing cells, and they are not able to activate the cytokine production by T cells. Second aspect, is it dependent on EWS5? Since the peptide is dependent on the neoproteins, which is dependent on the neogenes, which is dependent on EWS5, when we knock down EWS5, indeed, we lose almost completely, as shown there. Wow. I'm very bad at video games, as you can. So you see, when you knock down EWS5, you lose completely, almost completely, the cytokine production. And when you CRISPR-I the EWS and G8, you also completely lose this production. So those T cells can be activated by Ewing cells. Are they able to kill the Ewing cells? And this is shown here. So you have in the Y, this is annexin staining. So this is the amount of dying apoptotic cells across time after incubation between Ewing cells and CD8 T cells. And you see that all the Ewing cells with HLA-A2 can be indeed killed by the CD8. Ewing cells which are not HLA-A2 cannot be killed, and non-Ewing cells cannot be killed. This killing can be completely abrogated by a specific anti-class 1 antibody, showing that indeed this interaction on class 1 is key to this. And if now we do EWS5 knockdown, again, we lose. So we have a good knockdown of EWS5. We have then knockdown of NG8, and we considerably decrease the cytotoxicity of the T cells, again, Ewing cells. And if we CRISPR-I the NG8, we also observe with two different SG RNA, we also considerably decrease this killing. So we believe that this is quite important because we have here some peptides which are completely specific for Ewing cells, not observed in any normal tissue on the organism, sorry, which could be a fantastic target for immunotherapy approaches. One could be vaccination, to vaccinate with those peptides, to boost somehow the immune system to be able to recognize cells with this peptide on an HLA context. It could be also bispecific T cell enhancer that bridge Ewing cells and T cells, or TCR T cells by manipulating T cells from the patients in vitro with the appropriate T cell receptor able to recognize it, and re-injecting with, you may know some success of TCR T cell therapy, particularly in synovial sarcoma with NY-ESO and this kind of antigen. And I come back to my introduction here, which I showed you the many different transplantations that are observed in patients. So maybe the other translocation, the abnormal transcription factor, also lead to some kind of expression of neogenes. And indeed, we took a number of different types of such sarcoma, and indeed, for all of them, we could detect in red here, very specific sets of neogenes, very specific for each type of abnormal transcription factors. So meaning that what we observe for Ewing sarcoma linked to the microsatellite can also be observed for some other tumors, even though we don't know what would be the mediator on the DNA of the activity of those other transcription factors. But we believe that this is the transcription factor that is very important. Just show you this example, is an example for pathologists mostly, two tumors that are quite different. I'm speaking under the control of those guys, like the angiofibromatoid histiocytoma and clear cell sarcoma, two different tumors, but that have the same fusion, express the same set, as shown here, the same set of neogenes. So really meaning that the translocation is key to this induction. Okay, and to conclude with this potential vaccination of T-cell therapy in Ewing sarcoma, I would like to allude to clinical trials that is ongoing and which is a very raw vaccination. The idea is just to take Ewing cells from the patients, grow them in vitro, increase the expression of HLA-A2, HLA class 1 molecule on the surface, then irradiate those cells and re-inject to the patient. And this is interesting because they observe some immune response and they also observe some clinical response with this very raw approach to vaccination. But that means that in Ewing cells, even though there are very little mutations, there are some antigens that can be recognized by the immune system and can attack the cells. So maybe the neogenes I told you about are those guys. And with this, I think I will conclude, thanking all my colleagues, so Julien and Olivier, all the work on the neogenes, and then Céline and Florian, all the work on the peptides, and then the immunology in close collaboration with the group of Olivier Lance in Institut Curie, and a very important collaboration with Stephen Shannock at the NCI on the genome-wide association study, and with Barcelona for their fantastic immunopeptidomic platform. And I thank you very much for your attention.